Hello, this is Mr. Anger again with another coffee talk without the coffee breath from Biology Pace 1107. This is genetics, and we're going to be talking about the section that picks up after the first checkup. And let's see, it begins on page I in the Pace, and if you have read that, or somewhat familiar with it, then let's talk about some of the terms and try to make it a little more useful to you. You read about a man named Gregor Mendel. Gregor Mendel lived in what is now Czech Republic and was a monk, but he had a garden and he did a lot of experiments with his plants, and particularly he did a lot of gardening with pea plants. And notice that there were certain characteristics, like whether they were yellow or green, or whether the plants themselves ended up being tall plants or ended up being short plants. Or, here's another one, whether the peas were round or whether the peas came out wrinkled. And what he noticed was that if he had a characteristic, and your Pace goes through a whole long example with the yellow versus green, so I'm going to take a different example of the round versus the wrinkled pea plants. And he noticed that if he kept crossbreeding the plants or breeding the plants over and over again where the peas were always round, always round, always round, and in another part of the garden he had plants that the peas always came out wrinkled. Then if he took those two plants from the two different parts of the garden and he crossbred them, in other words, he took the pollen from the one and he put the pollen on the pistils of the flowers from the other pea plants. And then he would notice, well, what did all of the peas in the new offspring look like? And it was very interesting because all of the offspring in that situation came out round. So he did the experiment again, but this time he took the offspring from that generation of plants and he just crossbred them among each other, and something very unusual happened, but very predictable, he discovered. And that was that 3/4 of the plants produced round peas and 1/4 produced wrinkled peas. He began doing a lot of thinking, trying this with other types of characteristics to see if he could come up with similar results, and he did over and over again with different characteristics. And he determined that there were, as he or someone later called them, alleles, which would be the characteristics that, and his theory was that there were two coming together in each plant, and that one must be dominant over the other one. So if they both appeared, then one would kind of like win out over the recessive one, and that would be the trait that would be characteristic. Now let's go back and talk just for a minute about what's inside. Remember, inside the nucleus we have the chromosomes, and there could be a chromosome coming from the one plant in this case and a chromosome coming from another plant, and at a particular point along that chromosome would be a gene. So let's say the gene right between here and here is the one that determines whether the peas are going to be round or wrinkled. And then on the other chromosome, there is the same gene. So we have two chromosomes paired together, having exactly the same genes, but on this one strand, it might dictate that the peas are going to be round. I'm going to use capital R for round. But on this one, it might dictate that they would be wrinkled. So I used a little r. The small letters indicate the recessive trait. And what he figured out is that if both of those alleles are present, then the one that's dominant will win out. So in this case, with these particular chromosomes and genes, this plant is going to produce round peas. Well, a man named Punnett came up with a way of predicting outcome and using what's called the Punnett squares. And so let's talk about the first generation of plants that Gregor Mendel came up with using the round and wrinkled peas. And we're going to let, again, the capital letter represent the pollen from the round plant. And then we're going to let little r represent the plants from the wrinkled variety. And when he crossbred these two together, we end up with a capital R, little r. Capital R, little r. I'm taking here from here and here. And then down here, I take this letter and this letter, put them together. And notice that all of the possibilities for the offspring are exactly the same. Big R, little r. The capital R tells us, again, that the dominant trait, which in this case is the round peas, is what's going to be evident. So all these plants produced round peas. We call this the F1, or the first filial generation. The first generation of plants from this crossbreeding. And then what Gregor Mendel did, remember, is he took some of the plants from here and crossbred them. So he did a capital R, little r, capital R, little r, to see what the offspring would be like. And this ends up being capital R, capital R. This one is capital R, little r, capital R, little r. But then here is little r, little r. And the little letters, the lowercase letters, represent the recessive trait, which in this case is, like we were talking about, the wrinkled peas. So out of all the plants, he didn't just plant 4 plants. I mean, there are lots of plants. But a ratio of 3 to 1, or 3/4, 75% of the plants ended up looking like they had the round peas, and only 1/4 had the wrinkled peas. Now let's go back and introduce some new terms that we need to know that kind of fit in with what's happening here. When we have, let me get an eraser here and get rid of this picture. When we have both traits the same, both alleles are the same, we call that homozygous. Same. Okay? So the homozygous, this would be homozygous for the trait of being round. This little r is homozygous for the trait of being the wrinkled. But then the other is what we call heterozygous, which means different. So when we have the capital letter and the small letter together, as we do in all of these, and in this plant versus this plant, heterozygous means the two alleles are different. And in a case like that, the dominant trait is the one that is demonstrated, the one that is visible and you can see. We can't really see at the genetic level. We can't see what's happening in the chromosomes. We have to do experiments and look at the outcome, and then we can go back and figure out what the actual genes would be. There's another term that's important, and that is phenotype versus genotype. Now in the root here of pheno, we have the pH, which reminds us of physical. And then here we have the genes, which reminds us of the genes or the genetic makeup of the plant. Like I said, we can't see the genotype of a plant, but if we play with what we think are the genes, we can put the genotypes up here and see whether it's going to be a heterozygous or homozygous for a particular trait. But the only thing that you can actually see or detect with your eyes and with your senses would be the phenotype. So here, let's talk about this now using these correct terms. We're taking a plant that's homozygous round with a plant that's homozygous wrinkled, and when we crossbreed them in the first filial generation, we have offspring that has the phenotype, okay, of all being round. But they also all have the genotype of being heterozygous because they have one of each of the alleles. Then, if we take one of these heterozygous round plants and crossbreed it with another heterozygous round plant, we end up with a variety here of genotypes. Here we actually have a homozygous round plant, and then another heterozygous and another heterozygous. We have two heterozygous round and one homozygous wrinkled. Are you following me? How many of these offspring, what percentage of them are round? 1, 2, 3 will appear round. Only 25% will appear wrinkled. That's the phenotype. But when we start talking about the genotypes, 25% is homozygous round, 50%, 2 out of 4, is heterozygous round. And then we only have the one that's homozygous wrinkled. Now here's a very important point that you need to understand. In order for any recessive trait to be visible or evident, there cannot be a dominant trait with that. They both have to be homozygous for that recessive trait. There are several traits that humans have, such as the ability to roll your tongue, like that. Whether the earlobe is attached or whether it's kind of lobed is a difference. Whether the first finger is longer here, the pointer finger, or whether the ring finger is longer. I believe mine is actually recessive. I think in most people, the ring finger is a little bit taller than the pointer finger, which means that for me to have my fingers appear this way, and my toes as well, that I must have the recessive trait coming from both of my parents for that particular trait, because I'm exhibiting the phenotype of the recessive trait. Only way that can happen is for it to be homozygous. So let's look at these terms again then, remembering alleles, each of these individually are called an allele, all right? When you put two alleles together, if they are for the same trait, then we say it's homozygous. If one's recessive, one's dominant, and you put the two together, then we say it's heterozygous. Again, the phenotype is what we actually can see or detect as a result of observations and experiments, and the genotype is what we can, through experimentation, figure out that the alleles must be. The first time we breed plants together, we call that the first filial generation, and then the second generation is called the second filial generation. And these type of experiments can be repeated for lots of different traits, not just round versus wrinkled. It could be tall versus short, yellow versus green. And actually the yellow plant, I believe, is dominant, and the green is recessive. Now we always think of pea plants as being green, but the more yellowish color is the dominant trait. Now that's where it gets kind of tricky, and you got to be very careful when you do this in the PACE, because when you're just writing Y's, it's real easy to make a capital Y and a small y look the same. So you might want to instead make the capital Y like that, and maybe make the small y with a hook on it or something, but you need to distinguish between them. And same for short. I think S for short is the dominant, if I remember right, and then so a small s would be the recessive, but again, that's really hard to keep those two straight. So I sometimes recommend when students are going through the section where they're comparing the short and the tall traits to just use S and T. Technically, we're supposed to use capital letter and lowercase letter, but it may be a good idea just to keep it straight and make it easier to score to use two different letters like S and T. So I hope this helps you, and as we've tried to explain the terms across from each other, and what they mean, and give you another illustration, a little different than the one that you'll read about in your PACE. Then now as you go and actually do the homework in your PACE, you can answer the questions better and do the Punnett squares a little better. And think about those questions as you answer them. Thank you.